I will share my screen again. And okay, guys, so most of the time we talked about longevity genes, and as well as normal genes, as well as damaging genes. And with practical examples, you saw that there is not everything well in my genome. There are some damaging and pathogenic variants. And in this workshop, bonus workshop, we will talk what can be done if you are unhappy about some mutations. It's a little bit futuristic because you probably want to wait a little bit when the technologies will get more safer and easier to use, but still actually you can pretty easily design gene editing tools, and you can pretty easily pack it in viral vector, and that's what I will share now. So when we are unhappy about our damaging gene variants, we want to fix them with good ones using different gene editing tools. And of course, different types of CRISPRs are the most popular ones. And but please, when you need a doctor to fix your genes, don't go to this doctor. So on the picture is Jiankui, the guy who applied the Cas9 on 2 twins. Actually, I believe it was really brave of him, but he did a terrible job by using wild type Cas9 that could cause additional mutations and slightly hurt the health. So let's first look a little bit at the theory. And we all know that for CRISPR-Cas9, there was a Nobel Prize awarded to Dudna and her collaborators. And originally, just to remind you that it was part of immune system of bacteria, and they worked with Streptococcus that is slightly pathogenic. And the main idea is that you have bacteriophages that, like on this picture, that get into the bacteria, and they have DNA. And the idea of CRISPR is that it can recognize foreign DNA and cut the hell out of it. And you have those CRISPR arrays, and they use RNA as the guide. So it unveins the DNA, matches RNA if it fits. If it fits, it does the cutting. And people, after people moved the system to use it for gene editing on mammalian cells, originally it was just knockdowns when you just make cuts and destroy genes. Then they utilized homology and non-homology directed repair to give a DNA template and make an insertion of the transgene. And actually, even though many labs, including some labs in our institute, are using this technology, it's already somewhat outdated. So using homology-directed repair for insertion is actually not the very best technique now, surprisingly. Several years ago, it was really hot. And if we look into CRISPR and gene editing overall, so the idea that you can program some protein to get into specific region and do the cutting is not new. People use talents, people use zinc fingers motifs, And actually, TALEN is still more accurate than CRISPR, but because you had to do changes in the proteins, it was a headache to use. And when CRISPR emerged, everybody switched to CRISPR. And as you can see from this gene editing trials, you see that 63% of them are done with CRISPR because you don't change protein, you just change RNA. And the other important stuff is that mostly they prefer to edit cells because you have a problem of delivery. So you see like in vivo gene editing and ex vivo gene editing. And ex vivo means that we take patient cells, we do editing, maybe we want to grow them, and then we put them back and cells do the job. And it's way safer and way easier to do. So most of the trials, as you see, are ex vivo. And also, you also see that most of ex vivo, like 68%, are CAR T cells. So it's T cells that we want to reprogram to target cancer. So they bind to cancer cells and kill them. And it's for some types of cancer, it's the best therapy ever, especially non-soluble cancers. And in terms of Romania, by the way, I think in Fundeni, they're doing some experiments, and also in several other hospitals and labs with CAR T cells. In our lab, we worked with other types of cells. So we had several cell lines that we actually edited for research purposes. And yes, so you see that the CRISPR is actually leader, and all the time new types of CRISPRs and new modifications emerge. And what actually he did in that famous human germline editing, for which he got actually a sentence. I don't remember, I think like 1 or 2 years of jail time, and he is on freedom again. So he knocked down CCR5 gene in Lulu and Nana, and they should be 3 years old by now. At least they were healthy. I don't know if they're still healthy now. Probably they are, because the chances are not that high that huge damage was made. And official version was that he wanted to make it protective against HIV. And actually, I personally think he is a bit smarter. He had also another intention. And regarding the mistakes that he made, scientists probably would say it is like using wild type Cas9 that could have off targets, and also not only like it can miss and cut the wrong gene because the guide can partially match DNA in another place. Also by itself, if you actually like in the institute, I know some people experimenting putting Cas9 without any guides, and if it doesn't have any guides at all, it very rarely, but sometimes actually cuts just for fun, and it's not good. And for this reason, people created a high fidelity Cas9 and also enhanced Cas9, and 3 years they were already available, and he should have actually used them. But the main mistake was that he got funding from wrong guys who were not friends of Xi Jinping, and when they spoiled the relationship, he made him go to jail. And there were other issues. So when you do stuff, you also can get the organism mosaic, like not all the chromosomes and all the cells got the edit. And the other thing is that actually he didn't save them from a serious illness. Maybe if it was a serious disease that he would fix with Indel, maybe society would be more nice to him. And also, like any protein, CCR5 has both pros and cons. So it is entry receptor for HIV, but also it is a bit protective for some diseases. So first, what you do is you typically look at gene cards. That, like, if you have a gene, well, I mean, there are many sources, like you can look Wikipedia, you can look G cards. So if you want to edit a gene, you should check attentively what is there. And the second thing, what you really need is you also check for existing SNPs of the genes, if there are any associations. So for the benefit of this tutorial, let's try to actually reproduce CCR5 edit. And actually, when you Google the gene, you actually realize that, well, it's not that good in terms of HIV protection. It doesn't like give 100% protection, but actually there is something that he preferred not to mention, but everybody knew, is that CCR5 is also a gene for some reason associated with enhanced learning, low hepatotoxicity, and is a bit protective against atherosclerosis. If you go so there is a database made by George Lab where he collects so-called superhuman gene, the genes that are super good for something. And he has like a blog that he found on human studies, and also he checks if those genes were really good for something in animals. And he also had specific part for cognitive related gene therapies, because actually that's what we humans love to do, to make our children and ourselves smarter. And in case of this gene, you can see that CCR5, on animal models, it was had enhanced learning, but it's not only on animals. There were studies on humans that actually proved that it's also good for cortical plasticity on humans. And the major problem, I believe, is that because of one bad old story with one German guy, things about human enhancement are sometimes treated badly by public media. And that's why instead of just publicly telling, I want to make kids smarter, he had to focus on another good property of the gene about HIV protection. And so let's make first the ugly interventions. So this gene, actually, he just broke it. And what I suggest is, so there are like 50 different tools to do it, because it's very simple to, because it's very simple to do the edit. Many people implemented the tools. And where is the chat? So please, let's just check, for example, Syntago, but you can check any other online tool. I give this link. If I will find where my chat is hidden, I will also copy paste it there. And let me actually do it as well. So first, what you do, you select Homo sapiens as a genome, then you do CCR5, and you do search. And so usually for CRISPR, you need to have, if it is Cas9, it has, it should be like 20 nucleotides long guide RNA. And before it, you should have a motif. So it should be like NGG. So any letter, NGG. And that's why so here they showed by default recommended guides, but we can actually see all the guides. So if you look into the guide, actually, I think it's 19 nucleotides. And before, so we have these 19 nucleotides. And in the genome, before these nucleotides, you have a motif like any nucleotide and GG. And that's why, and for the scores, they give scores, usually it's several popular algorithms like Doench algorithm, like Azimuth. I think Syntego uses Azimuth. It is a model trained with Microsoft. I think they had something like random forests, and it looks into the sequence and flanking sequences. So 19 nucleotide guide and 10 nucleotide sequence on the left and right. And here you get the scores. So you see how should it cut on target score, and off targets, the format is like 0 direct off targets, 0 off targets with 1 nucleotide difference, and 2 off targets with 3. And it suggested you to select 3 best guides. And also they looked into genes. So in genes you have different regions, like promoter, it's like start. Then if somebody is asking, please do in written form.
Non, c'est pas de l'eau, c'est plutôt.
And mute French people. I don't understand French, unfortunately. And so far, so it's coding region, the regions that will turn into proteins. It's in exons, and that's why we focus on destroying the exon. And usually with indel, we want to make a frame shift or to make a stop codon. And so here we have the guides, and we actually have to put it in a viral vector. And what about the delivery? So there are different ways. If you do on cells, easiest is electroporation, and there are very cheap designs for in vivo electroporators. But as Ukrainian, at least according to Russian propaganda, I should be good with bioweapons. I would prefer to put it in a viral vector. And to put it in a viral vector, you have to use a service. So I will put a link to the chat. I personally use Vector Builder because they have nice cloning and viral vectors production services. In terms of viruses, there are many of them. The most popular ones, let me make it larger, and also can also open it yourself with the link, are AIV. It's adeno-associated virus. It's the safest one, but actually it's of very low efficiency. We tested them on cells in the lab, and we did optimization, like selecting the best serotype. So the serotype is actually those spikes that interact in fibers that interact with the cell and provides the entry. And from our experience, adenovirus was much better. and it doesn't integrate into genome. So it delivers as RNA and transcribes and translates the protein of your choice. People also, quite popular choice is lentivirus, but lentivirus integrates into DNA. And if we want to reproduce what's like gene editing, say on humans, we probably don't want additional integration of some lentiviral part. And they actually have vectors for CRISPR delivery. So that's why I'm actually checking mammalian CRISPR gene editing vector. And what we need to deliver both Cas9 that will do the cut and guide RNA. So I select adenoviral. So starting price is $269. And they have also really useful guide to show how it works, what are the pros and cons. So here I will just go to design. And here I have a choice. So actually from the research, it's recommended to use 2 or 3 guide RNAs to for sure knock the gene down. So I select dual gRNA vector. And here I have the design. So what you see here is a plasmid map, and it's a circular DNA. And this is used to produce, so you put it to some cells, like it can be, usually people use HEK cells to produce adenovirus. And what is important for us, we don't really care about most of this part. It's like technical, like origin replication, but we have regions that will be transcribed to RNA. So if we want to get RNA expressed from the plasmid, we use U6 promoter. So promoter just tells that please read something after me into RNA usually. And let's return to where we selected the guides. I believe I had this window somewhere because we have to copy them. Maybe it's better for me to close redundant papers. So I had the, yeah. So let's select best guides. This one looks really good. And the first one will be probably, wow, it's Zoom thing is really invasive. Okay. Okay, so the first one probably be the highest score. So hello world RNA. And for the sequence, we actually it already did for us because it was in RNA format. In RNA format, you have uracil instead of thymine. And when I copy pasted, the vector builder actually figured out it and made the substitution. And what it gives to me, so here is my guide RNA. And also to make CRISPR work, I have a technical scaffold that is required to attach it to the protein. Luckily for me, in the plasmid, they already have it inserted. And that means that the only thing that I have to do is saying I'm happy with this design, add it to vector. And then I need the second one. I can also, I can copy any of this. Usually, like if you have on target more than 0.5, it's usually reasonably good. And let me like insert inserted. No, it's will be like second guide. And I'm happy for the vector. And I need to so they have like official name of this. So they already use this guide probably. Okay, so I have to select proper CRISPR, and I actually, so what they suggest is, so H means human, but it's just Cas9 that they codon optimized for human, just not really important. And they also added some text for purification that we don't really care about, and nuclear localization signals that just helps it to get into the nucleus. So two other options is, so here you have again CRISPR, but you have these two things. So in plasmid construction, you usually separate with, usually actually you do it with dash, not with colon, but here they did it with colon. So you have three elements here. Human Cas9, then you have P2A. P2A is just self-cleaving peptide. It means that if you want to make a trick to express several proteins from the same gene in mammalian cells, you cannot do just, you need, usually it's one promoter and one protein expressed, but if you put this P2A and then put second protein, it will express all this first as one protein and then will cut them here into two. And the third part is just glowing. That means that it gives CRISPR and it gives glowing. And if we want to do some debugging, like to see if our child gets most of the cell got CRISPR, we would probably prefer to also have some glowing for debugging purposes. Although it's not really recommended. It's better people debug on cells, and when they go in vivo, they go without glowing because it can a little bit damaging. So we got our vector, we push finish design, and we finally get, why is it taking so long? Okay, so nice. We got it. It will be produced, so it will take 20 days for them to prepare and $369. But we have problems that it's only like the DNA that they are making for us. And we have to actually add the packaging into viral particles. So we have to add this downstream service. And it means it will cost us $600 more approximately. And it takes, I think, around 20 extra days from 20 to 40 extra days to make those viral particles. So we will be able to just inject them to wherever we want. And that's if we that's how things work for CRISPR viral delivery. And coming back, this panel is so annoying.
So, Anton, sorry for interrupting me, but we don't have more time. Please try to.
Okay, so yeah, so we finished Hello World, and then what is important actually here is that, I mean, I showed you not the most optimal way because AD5 is somewhat immunogenic and you have to actually use a bit modified viruses in vivo, and overall you should wait maybe 5-10 years when it will be useful and safe on humans. And also some people, it's also important, don't try it on yourself until at least you will check that you don't have immunity, because Streptococcus is immunogenic, and its Cas9 can, in 20% people, can cause immune reaction. And actually, it's the ugly way of doing it because the technology is pretty bad. And what I would suggest doing, so if we just want this receptor not to be expressed, we actually don't really need to kill it and to alter our genome, because we can do we can use epigenetic CRISPR. So what people did, they mutated, they created dead Cas9, so they broke cutting ability, so it can only sit to DNA and do nothing. And the second thing is that they added modifiers to this protein that can, in this place of DNA, do some job. It can be activation, suppression, or if you put methylation, it will memorize this methylation, and this gene will not be active, will not be expressed for more than 50 days at least. And it's the way minimum people didn't test years. But if we want to do it with epigenetic CRISPR, it's pretty much similar. The major difference, I don't have time to go through these details, but it should be near start of transcription. Start when you, after promoter, you start transcribing DNA to RNA. If you put it somewhere close, plus minus 500 nucleotides, it will silence this gene and it will be heritable, and it can be cancelable by another epigenetic CRISPR or other mechanism if you want. And I don't have time, but there is CRISPR era and other services to, you just use services for inhibiting CRISPR. And then, because it's not on vector builder, you go to add gene, and you have to get this plasmid and send it to vector builder for further cloning, because vector builder, or you have to synthesize customly because they don't have template for epigenetic CRISPR. And the last thing is the proper editing, and the proper editing is actually CRISPR prime editing. Again, it's, I don't have time to go into details, but the idea is that you use nicases that cut only one strand of DNA, and you use a very tricky PEG RNA. That means that, so they've used that Cas9 with reverse transcription, that you give RNA to go to appropriate place, and you also give part of RNA that will be immediately turned into DNA. And because of very partial one-stranded mismatch repair, that is not really dangerous in comparison with double-stranded DNA repair, it will allow incorporating the sequence you want, and it's a bit less efficient, but it is much more safer than regular CRISPR HDR editing. And people even investigated and the stuff and improved it with combination with integrases. You can get inserted part with integrase site, and integrase a protein for actually originally also used in integrating phage DNA into bacteria, and it was repurposed, and you can get like 20,000 nucleotides inserted wherever you want. That is pretty cool. I give links to the papers, and this is comparison with HDR. So you can see that indels, the kind of dangerous guys. So it has very, so HDR is a bit more efficient than PASTE, but number of indels is crazy. So it's better to do PASTE multiple time than to use regular HDR. And that's the takeaway with CRISPR. And now you have learned how also to edit the genome. And thank you for your attention, and happy for your questions.
I think there are questions in chat, and I don't know how much time we have left, but if you may answer them, that would be awesome.
Yeah, Anton, we have maximum 5-10 minutes.
Yeah, so so the service, I think you have to like I mean, when you deliver in dry ice, actually, like every person cannot get like account in dry ice delivery. Usually they check who you are. So you can quite easily get design and get quote, but like if you want to deliver home, you can have issues with delivery, and delivery can cost a lot. Like dry ice delivery can cost like $1,000 easily. It's a crazy the way how prices are made in dry ice delivery. For Liz Parrish paper, yeah, I saw it. They use another type of virus. It was, I think it was leukemia M something virus. They used the delivery through ice. So actually, it's quite common to deliver viruses through nose or ice. I don't remember. I think it was nose actually, nasal injections. Yes, so and they actually had the H3, and they had a second gene that is follistatin or something. My memory is pretty bad. For those people who did CRISPR on themselves, so the question how efficient it was, actually it's not that super easy to deliver it efficiently. The easiest thing is if you want just to deliver to skin, like for example, you want to make glowing skin, like you deliver GFP, you do in vivo electroporation. It's quite painful. There are open source designs that you can just within $100 for consumables actually assemble this electroporator, but it will first damage your skin. Second, you have to check if you don't have autoimmunity against Cas9, otherwise you will be in a big trouble with adverse reaction. And third, I would actually recommend always to try on cells or animals, because then on yourself. What else? I know, I think it's like all questions. I don't see any other questions.
Wait, what do you mean try somewhere? You received this CRISPR. Can you multiply it to use?
The thing is that there are different ways of delivery. So I don't recommend to use viruses, at least the method I showed to you. Like adenovirus is highly immunogenic. Even if you use gutless adenovirus, better don't try it on yourself. So if you get viral particles, you can inject them, but they have mutations that don't allow to multiply them. So you need helper plasmid to multiply. So you cannot, you get some number of particles, but you cannot multiply them unless you will put helper plasmid, and you will have a cell line that will be producing. For things like injecting, like electroporations that you can do probably with skin, there you actually don't need a virus. You just need to buy, it's pretty easy to get some plasmid DNA, but efficiency, I don't know. I mean, you will probably reach only to part of the skin cells. It's a common problem in vivo, and you will hurt your skin because of electricity. And I don't have expertise with in vivo electroporation to say how many cells will get CRISPR. And it's of a limited use. I mean, okay, you modify part of your skin that you also damage with electricity. So what stuff you get? What's useful stuff you can get at this small patch of skin?
That was the question. Are there any useful use cases?
Well, I mean, you probably can deliver elastin. So because usually it goes down with age, but maybe you can deliver some regenerative things. But the problem also is that it will not be really specific. And if you have different cells, like for example, elastin can be good for skin and arteries, but I don't think like neurons will benefit much from elastin. So I mean, things are complex, and even though it's not that super hard to do something stupid, in most of the cases, it will just not work.
So the most people who order this on this paid services, they just do some experiments.
Yeah, most of them are because you can spend a lot of money to make some fun, but usually you kind of have to plan really hard and experiment a lot to get results. So maybe you can do something on your kitchen, but the easiest thing is just to make things glowing. But okay, you made it glowing, but what is the applicability of it? Go to a party and show? Maybe that's it. And for regeneration, it's hard. You have to do a lot of research. Like you can deliver the factors, but you have to understand where, how. And so the stuff I gave you is just for fun. I thought maybe.
Okay.
Okay, guys, so I see no more questions. So so far, every of you will, question. Okay, Arseny, you will be the last one. Larissa needs to sleep, Okay, Arseny, last question. So Vector Builder is in China, actually. Actually, in Europe, it's a limited number of companies that you can deal with. But in Vector Builder, things go from China, and it's actually a big headache because of transportation. In European Union, there is a viral core in Barcelona, but you have to wait 6 months until they will do anything. Really annoying. Okay, guys, I finished, so maybe we can close it.
So I would like to thank our speakers. Thanks, Anton, and thanks to your team and Kim. And also, I'd like to thank our participants, and I'm looking forward to see you again to our next webinars.